excited to be here, and today I want to talk to you about Rubisco in marine environments. And so you've had a little bit of introduction to Rubisco, and Rubisco is the enzyme that fixes carbon dioxide in photosynthesis. So just to put it into context, I'm going to go straight to this slide. This is a schematic of how photosynthesis works, and you can see Rubisco up here. This is the enzyme that takes CO2 and fixes it into organic carbon. It is used in oxygenic photosynthesis, so all plants, algae, cyanobacteria all use this process here, and you can see Rubisco is part of the light-independent reaction, and it gets the reductant and energy to power this light-independent reaction by using this light-dependent reaction, which harnesses the sun's energy and uses chlorophyll and all that to generate oxygen. And so, as I said, all plants, algae, cyanobacteria use oxygenic photosynthesis to autotrophically fix carbon. And this is by far and away the most dominant way of fixing carbon on our planet right now. But there are parts of the Earth which are, there is no light, parts of the early Earth where, and different environments where oxygenic photosynthesis wasn't around. And there's actually plenty of bacteria and archaea at the moment that also use Rubisco. But they don't have this light-dependent pathway. They can oxidize other chemicals and do other reactions to generate reductant and energy. So I really want to focus on Rubisco because it is such an important part. When you think about how carbon is fixed on our planet, Rubisco is the main one that does that. And I want to talk about it in the marine environment because half of all the photosynthesis on the planet happens in the oceans. All right? So that means that every second breath you take, that oxygen was produced by algae in the oceans, which is so cool. And in the oceans, the oxygenic photosynthesis is largely produced by it's this group called phytoplankton, which is a very loose term that covers all sorts of single-celled microbes that do oxygenic photosynthesis. And they come from a remarkably diverse background, all the way from cyanobacteria to sort of heteroconts, which are diatoms and coccolithophores that make these beautiful shells, there's green algae. Anyway, it's just incredibly diverse. They have all these different types of Rubisco compared to plants, which all arise from one ancestor with, one type of Rubisco. So the diversity in the oceans is huge. And then you go into the deep oceans and hydrothermal vents and anoxic zones, and you have all the bacteria and archaea that are also using Rubisco to fix CO2. So if we want to understand how Rubisco has evolved and autotrophic carbon fixation has evolved on our planet, the oceans is a great place to look. And so Rubisco stands for ribulose 1,5-biphosphate carboxylase oxygenase, and I'm getting very good at saying that. It's an amazing enzyme, right? So I said 99% of all biologically fixed carbon is thought to go through this one enzyme. They think it's ancient, over 3 billion years old, and you would think with over 3 billion years of evolution, it would be very well adapted and good at what it does right now, right? But it's not. It is terrible. It is a terrible, terrible enzyme. It is really big. It is really slow. You need a high concentration of CO2 around Rubisco for it to work. And it is competitively inhibited by oxygen, which is a challenge, right, in an oxygenated atmosphere. And so the big question is why is this the enzyme of choice? Why is this how this planet fixes its carbon for life? And so I want to introduce to you a couple of projects that my lab group is going through studying Rubisco and the diversity of Rubisco kinetics in modern species and how that can help us inform this question. And so I want to say that one of the fundamental sort of the current consensus of why Rubisco isn't so much better than what it is because Rubisco is adapting, it is evolving, but it is biochemically constrained between how fast it can fix carbon and how specific it is for CO2. And so what I'm showing you on the left-hand panel here is basically the speed of carboxylation, which is the kcat, against its specificity for CO2 over oxygen. And you can see they generally fall across this line, that the faster you can fix carbon, the less specific you are for CO2. Now the problem is most of this data is based on plants, because that's what people study. It's really easy to go out and grab a plant. These two outliers, these are cyanobacteria. All right. And actually, if we go into the marine environment and extract Rubiscos from a variety of different organisms and measure their Rubisco kinetics, we see a remarkable diversity. So that's the middle panel here. And what I'm showing you on the y-axis is the Kc. This is the half saturation constant for CO2. So the concentration of CO2 you need to give half the maximal rate. So a high Km means that you need a lot of CO2 to saturate the enzyme. A low Km means has a good affinity for CO2. And then these are the values from bacteria, diatoms, which are a really dominant phytoplankton group in the oceans, some red algae, the plants, and the cyanobacteria. And you can see this variable response. And I want to point out that this is a log scale on the y-axis, all right? So there's a truly dramatic range of Rubisco kinetics in these different organisms. And if we take these diatoms, which are dominant in the marine environment, and plot their Kms and carboxylation speeds on where we would expect them to fall, so the gray is the expected relationship between carboxylation speed and affinity for CO2, and the diatoms are in black here, and you can see they fall completely not on the line. All right, so we have a fundamental misunderstanding of how Rubisco works on our planet. And the other thing is that these measurements are made at 25 degrees Celsius, which is very relevant for plants, but not so relevant for the marine environment. And so in the marine environment, we get, oh, sorry, don't need the sound. In the marine environment, we have, sorry. All right, you're just gonna have to listen to it. This is the GoPro going through the sea ice up in northern Alaska. And I don't know if you can see it very well, but this brown coloration on the bottom, this is 1.5 meter thick sea ice. These are all diatoms growing within the bottom 5 centimeters of sea ice, right? It's an incredibly productive ecosystem. It feeds everything up there. These are microscopic organisms that just grow in these dense mats. And this is at -2 degrees Celsius. So we know that temperature has a big effect on how enzymes work, and Rubisco is no exception. And from what we can see, extracting Rubisco from polar diatoms, from warm species, all these different things, it doesn't look like Rubisco is cold adapted, which means at cold temperatures, it slows down dramatically. So in pink here, I'm showing you the carboxylation rate of, which is on this axis here, at 20 degrees and at 0 degrees Celsius, and you get an 8-fold reduction of carbon fixation speed going from 20 to 0 degrees Celsius. All right, so it slows down a lot. In addition, the Km, the half saturation constant, goes from about 40, which is shown on the, it's in blue and it's shown on this axis here, goes from about 45 down to 15. So it becomes more specific for CO2, but it's really, really slow. And so polar diatoms at cold temperatures, to compensate for the slow carboxylation rate, they dramatically upregulate how much Rubisco they have. And so this is a polar diatom species here, grown in the lab, called Fragilariopsis cylindrus. This is data from the field in Antarctica of a big diatom bloom at -2 degrees Celsius. And they have about 16% of their total protein as Rubisco, compared to only about 2.5% at diatoms that grow at about 20 degrees Celsius. And this raises an interesting question on the theoretical how fast you can grow and fix carbon at cold temperatures, because it's a combination of how fast the Rubisco enzyme works and then how much Rubisco can you actually make in your cell. So it gives an interesting theoretical limit of how fast carbon can be fixed. And so this work is being continued on. I have 2 postdocs, Meng Li, who's actually starting on Monday. So we have all these Rubisco kinetic measurements over a range of temperatures. He will be using membrane inlet mass spectrometry to measure carbon fixation of the whole cell over these different environments to try and get a good idea of whole cell carbon fixation rates and Rubisco kinetics. And then Katrine Schmidt, who I'm not sure, she's not here today, but she is wandering around this week. She is growing sea ice in the lab that we can grow algae in the sea ice. So we get a lab environment that's much more representative of what we see out in the field. So another project I want to talk about is looking at Rubisco fractionation. So we just had a great talk on delta C13 and Rubisco fractionation. And yeah, so a lot of the assumptions are that Rubisco fractionates at 25 to 29 per mil. Once again, this is based largely on literature on plants at 25 degrees Celsius. And so what I'm showing you here is a phylogenetic tree of all the different types of Rubisco that are found in different organisms. I stole this tree from, you can't see properly, but it's Kajar Bettel's paper. So these are the different types of Rubisco. I've matched it up with the different organisms that have these different types of Rubisco. And so plants all have this form 1b. But in the marine environment, we actually have most of them, the eukaryotic phytoplankton, have form CD, right? So a lot of diatoms, coccolithophores, have this CD group. And then there's cyanobacteria, some have 1a, some have 1b. There's very few measurements that have been taken. The ones that have been measured and published, I've shown on the right-hand panel here. I want to reiterate, I forgot to put the references up. This is not my research. This is a compilation of literature by a whole bunch of different people. But I have the list. So if anyone wants a specific number, I can tell you who did the research and which paper it is. But what's interesting is we only have 3 measurements from marine eukaryotic phytoplankton, and they are hugely variable. EHUX has a value of 11 per mil, a diatom has 18, a red algae has 30. There's this dramatic difference of Rubisco fractionation from these different types of, all from form CD Rubisco. So we need to look into this a little bit further. So my grad student, Susan, who's in the audience, is working on extracting Rubisco and measuring the isotopic fractionation. So if you have any questions, definitely go talk to her. And the last thing I want to talk about today is looking at Rubisco in even more extreme environments than in sea ice. And so I want to go back to these cryopegs that Jodi Deming was talking about. And she already mentioned that we found Rubisco in there, so you've stolen my story, but yeah, it's... But these cryopegs are truly amazing environments, right? They are ancient marine sediments. For those that weren't here for Jodi's talk, but I think most people were, they are ancient marine sediments trapped within the permafrost. They are really cold, about -8 degrees Celsius. They're dark, they're anoxic. They're anywhere between 14 to 40,000 years old, right? And they are filled with bacteria. They are filled with organic carbon. So the vast majority of the bacteria in there are heterotrophs, but we wanted to see if there were Rubisco. And so once again, this work was done by Josie, who is Jodi Deming's postdoc. These are metagenomic samples from different cryopeg samples that they took. And these are Rubisco genes and their associated bacteria up here. So we see this wide diversity. They're not in high abundance, these genes, but they're definitely present in the cryopegs. Carbon fixation is happening. And one bug I wanted to pull out in particular, because this one kept coming up, is this Gammaproteobacteria called Thiomicrospira artica, which I stole this picture from this paper. It's an obligate chemolithoautotroph. It's sulfur oxidizing. It has 2 Rubiscos. It has a form 1 and a form 2. Now, lots of proteobacteria can have 2 different types of Rubisco, but generally, the form 1 is expressed under oxic environments. The form 2 is expressed under anoxic environments because they have different kinetics. Why this guy in this very stable ancient environment is keeping these 2 Rubiscos might be a really cool thing to look at to understand how gene evolution works in these extreme environments. So just to round it up, 99% of all biologically fixed carbon goes through Rubisco. It's this amazing ancient enzyme. And by studying the kinetics, the adaptation, the evolution, the diversity of Rubisco in all these different organisms and environments in the modern planet, we can get a really good idea of trying to reconstruct how evolution, adaptation, autotrophic carbon fixation has evolved on the planet. And just to say thank you, obviously this work was a big compilation of lots of people. So Hannah Dawson actually is my grad student. She gave a talk yesterday on sea ice algal metabolism using metabolomics. We have Susan and Katrine, the Deming Lab group, and these are my funding bodies. So yeah, thank you very much, and I'll take any questions.
Thank you, Jodi. Maybe one quick question?
Hey, I'm Tony Murray from Georgia Tech. So you said that there's a bunch that sort of lie on this nice trade-off line between speed and the specificity and whatnot, but there's these others that don't. Mm-hmm. And then you mentioned that temperature changes these numbers. Are you thinking that maybe ones that don't follow a nice trade-off might be trading off with something else, like not necessarily temperature, but something else, but another dimension that maybe the trade-off line is just a slice of a trade-off plane or something like that?
Yeah, no, it's a good question. I think there's obviously a lot more complexity there that we don't fully understand yet. The trade-off line I showed you, all those, that was all measurements taken at 25 degrees Celsius. So all those ones were like the standard when you measure Rubisco kinetics. It's 25, so you can compare everything. Yeah. But yeah, obviously they don't function at 25 degrees Celsius, all of them, and so they've evolved to different CO2 environments, to different temperatures, different salinities, all sorts of pH. You know, there's a whole myriad of different things we could look at, and it hasn't really been explored yet, so it's something really cool to look at.
Okay.
Yeah, thank you.
Okay, thank you so much.